Hello, and welcome to this video in the series on Integrated Pest Management, focusing on disease control in cereals. In this episode, we're going to take a wider perspective and look at the impact of disease control on the environment. Now, one view would be that disease control sometimes involves the use of fungicides, and pesticides are bad for the environment. But in this episode, we're going to take a look and explore whether that's the whole story. In this video on integrated pest management, we're addressing the question, is disease control good or bad for the environment? So, the major challenges for agriculture are to increase food production for a growing global population despite the challenges of climate change, and at the same time to reduce the environmental impacts of production. So here we're going to focus on 4 specific impacts related to disease control. So the impact of fungicides on non-target species, the effects of disease control on greenhouse gas emissions, land use and water use. We think first of all about the impacts on non-target species, including humans and a whole range of species in the environment. Then clearly there's potential for an effect on both biodiversity and species abundance. This is a huge topic, and I would recommend this publication from the European Environment Agency because it reviews the evidence in this area and provides a lot of links to information and research from what I would consider to be well-balanced sources. But if we think more broadly about the environmental impacts of production, we looked in the video about which parts of the crop canopy need protecting at this diagram, which shows the relationship between the amount of green canopy that we grow and the amount of energy from the sun that we're able to capture for photosynthesis to produce food. If we think about this diagram a bit more carefully, along the bottom axis, the amount of green canopy area that we produce is related to, first of all, cultivation, which involves burning diesel in order to establish the crop. It then involves nutrient applications, and canopy size is pretty much directly related to the amount of nitrogen uptake. And then we need water in order to maintain that canopy during the grain filling period. So those are all the inputs that allow us to grow the canopy and expand the green leaf area index along the bottom axis of this chart. And we do that because if we grow a bigger canopy, we're able to intercept more of the solar energy for photosynthesis and therefore produce more food. So we can actually consider this diagram to be the relationship between environmental impact and food production. So let's look at that in a bit more detail. This is a chart of the contribution of different inputs to wheat production and their impact on greenhouse gases associated with crop production. We can see, for example, on the right that the diesel involved in cultivation makes a significant contribution to greenhouse gases, but by far the biggest impact on greenhouse gases comes from nitrogen use. So the manufacture of nitrogen fertilizer through the Haber-Bosch process is hugely energy intensive. Then when we apply nitrogen to fields, we can stimulate the release of nitrous oxide, which is a particularly potent greenhouse gas, and that adds to the baseline nitrous oxide releases from the soil. Now, what's this got to do with disease control? Well, the interest of agriculture is that pretty much everything interacts with everything else in agricultural production, and one of the examples is the interaction between disease control and the effects of nitrogen. And this is an example from one set of field experiments where we measured the effect of the nitrogen fertilizer rate applied in kilograms per hectare on the yield of winter wheat. And we can see a number of things from this. The different curves represent different levels of disease control. So the bottom curve is from an untreated crop which was heavily diseased. As we control the disease more and more effectively, in this case with a more and more intensive fungicide program, we can see that the yield goes up and the response to nitrogen treatment also goes up, resulting in an increase in the economic optimum amount of nitrogen to apply. Now, there's a huge amount of variability between field experiments. If you repeat this experiment in different sites and seasons, you get quite different nitrogen optima and quite different yields. But the overriding and repeatable thing is that as we improve disease control and we optimize nitrogen fertilizer treatment, yield goes up. And that has pretty big implications. So in a publication back in 2008, we took data from the AHDB and looked at the impact of fungicide treatment on yield. And at that time, the average difference in yield between fungicide treated and untreated was just over 20%. Now, the implications of that for the amount of land area required to produce the amount of wheat to meet UK demand for consumption, which is about 15 million tonnes per annum, would equate to approximately 500,000 hectares of land. So if we just withdrew fungicide treatment and carried on doing everything else the same, we'd need about an extra 500,000 hectares to meet the demand. Or in the traditional units of land area, that's about 700,000 football pitches or a quarter of the land area of Wales. And the greenhouse gas implications of that are also very substantial, because the options then would be either that we move more land into arable production in order to meet the demand from domestic production, or we import and essentially export the land requirement. And just to take two examples of the consequences of that, if we chose to find the extra 500,000 hectares by converting grass to arable, then that would release about 6 tonnes of carbon dioxide equivalent per hectare per year. And carbon dioxide equivalent accounts for the relative heating effect of carbon dioxide, nitrous oxide and methane. If on the other hand we chose to export, as it were, the land requirement by importing more grain, and in the worst case that was achieved by cutting down forest in order to move it into arable production, then the impacts of that are about 20 tonnes of carbon dioxide equivalent per hectare per year. And we can contrast that against the benefits of having land returned to semi-natural environments, which is about just over 1 tonne of carbon dioxide equivalent sequestered into the soil per hectare per year of idle land. Now, this land use change effect has a big impact on how we optimize agriculture for greenhouse gas efficiency. And I'll just give an example on the effects on nitrogen fertilizer use. So here's a set of calculations, again published in 2008, where we looked at the effect of different contributions to greenhouse gas releases of different agricultural inputs. So at the bottom, operations to establish the crop, in green, the greenhouse gas costs associated with agrochemicals, then those associated with nitrogen fertilizer emissions and nitrous oxide emissions. If we ignore the effect of land use change, then the effect of all that would be that if we wanted to optimize greenhouse gases, we would actually apply virtually no nitrogen fertilizer. But as we've just seen, nitrogen fertilizer has a huge effect on the yield of crops and therefore the land use requirements. And when we account for the greenhouse gas effects of land use change, we can see that the optimum input of nitrogen for greenhouse gases actually comes closer to the economic optimum. So we took account of this in a more sophisticated analysis of the effects of disease control in a paper we published in 2010. So here we took account of the effects of disease control on the optimum nitrogen rate. We took account of land use change, and we made an assumption that the additional land required, if we reduced the level of disease control we obtained, would come from converting temperate grassland. And the outcome of that calculation was that if we failed to control diseases in wheat, then emissions would rise from 503 kilograms of carbon dioxide equivalent per tonne of grain to 713 kilograms of carbon dioxide equivalent per tonne of grain. And that would amount to just over 3 million tonnes of carbon dioxide equivalent per year to produce the domestic requirement of 15 million tonnes of grain. Now, the good news since 2010 is that the plant breeders have been busy improving wheat varieties. And we can see the effects of that if we look at the AHDB recommended lists. And in particular, if we look at the difference between the fungicide-treated and fungicide-untreated grain yields. And if we look at the quality wheats, which are shown here, or we look at the feed wheats, there are now varieties available in all of those categories where the difference in yield between treated and untreated is now around 10% rather than the 20% that we saw earlier. Now, these values vary quite a bit from year to year, but nevertheless there's been huge progress through plant breeding, and the effect of that has been to reduce the fungicide requirement of UK wheat crops that we need in order to achieve effective disease control and efficient crop production but nevertheless fungicide use has remained quite stubborn. So let's now think about water use. If we look at the leaf surface of winter wheat, it's covered in thousands of stomata, and these stomata are the openings in the leaf surface that regulate the exchange of gases from outside to inside the leaf. Now, we need to have gas coming into the leaf in order to fix carbon dioxide from the atmosphere in order to produce carbohydrate to produce grain. But in opening the leaf surface to allow carbon dioxide in, the leaf is also allowing water out. And despite the fact that the stomata regulate the gas exchange very carefully, for example by closing at night when there's no light available for photosynthesis, the amount of water lost is huge. Typically, although this varies quite a lot, the leaf will lose between 400 and 500 tons of water vapour for each ton of grain dry matter produced. And if we work that out for the 15 million tons of wheat production, the numbers are pretty staggering. So wheat crops are transpiring about 6 billion tons of water in order to produce that 15 million tons of wheat grain. And because arable crops form a major part of the land area, particularly in eastern England, this transpiration of water from the soil and putting it back into the atmosphere is a major contributor to why river flow in eastern England becomes very low or sometimes even stops during the peak summer months. So this water use is really important to the environment. Now, we might ask ourselves, well, what's that got to do with disease control? And it turns out quite a lot. In the left-hand image, which is a scanning electron microscope of the leaf surface, we can also see alongside the stoma a powdery mildew spore that's attempting to infect into the leaf surface. And in the right-hand image, we can see a couple of the effects of that attempted infection. So the little white spots on the right-hand diagram show where the mildew spore has tried to infect, and the plant leaf surface has responded in order to stop that infection. However, in some cases we can see on the left-hand side a little mildew spore that's been stained blue that's produced hyphae in order to infect a plant epidermal cell. And we can see in the white area in the middle of this image where the cell has died in order to prevent that infection in a reaction known as a hypersensitive response. So this is the plant disease resistance response, killing one of its own cells in order to prevent a mildew infection. And that has an impact on water use. So those disease resistance responses are associated with stomatal dysfunction, and that dysfunction can include stomata remaining locked open in the dark when they should be shut, and thus increasing the loss of water. And this has been shown in particular for resistance against powdery mildews. However, if we fail to control diseases, that's also bad, because many diseases impair stomatal opening in the light, which then interferes with carbon dioxide uptake. And some diseases themselves impair stomatal closing in the dark, for example, Rhincosporium in barley. And then we have the effects of the rust pathogens. And if you look very carefully at a rust pustule, you'll see, as in the image at the bottom, that it causes the surface of the leaf to burst open. And these openings are far bigger than the stomata, and they're obviously not regulated openings. So the water vapor from inside the leaf just pours out through these rust pustules. And the effects of that can be quite substantial. So this is data from a field trial where we had a susceptible variety that was heavily infected with yellow rust and a resistant variety, which was actually an isogenic line of the susceptible variety into which a resistance gene had been bred by traditional means and then by backcrossing. And then we superimposed on that either fungicide-treated or untreated plots. And we then took samples of the soil and measured the uptake of water by the crop that was caused by the transpiration we've just been talking about. So we measured the number of tons of water needed to produce 1 ton of grain. So in the susceptible yellow rust affected plots, that amounted to just below 400 tons in this case of water use per ton of grain. Whereas if we controlled the yellow rust, either by fungicide treatment or by the host resistance of the variety, that water use requirement went down by about a quarter, which is a very significant reduction. So what can we conclude from all of this? Most environmental impacts of cereal production are related to cultivation and nutrient use in order to establish and grow a crop canopy. If having made those investments and had that environmental impact, we allow diseases to destroy the crop canopy, then that's clearly very inefficient. So effective disease control reduces the land area that we need in order to meet the UK demand for food and feed. It increases nitrogen uptake and utilisation efficiency, and it reduces greenhouse gas emissions per tonne of grain, and also increases water use efficiency. Now we can obtain that disease control by a combination of integrated pest management methods, as we've seen in this series of videos, including through the use of disease-resistant varieties and by treatment with fungicide, and that treatment with fungicide should be according to need. And if we're thinking about the environmental effects of fungicides, we should take, I think, a broader view and balance the environmental impacts of fungicide against the environmental benefits of disease control. And if you want to read further about this subject and look at the evidence, there's plenty of information to go at in this further reading.